This is a picture that just kind of shows us what protein synthesis is. I'll come back to this near the end when it hopefully will make a bit more sense. And what we're going to talk about now is exactly how we build those proteins out of the genetic information found in DNA. And this is a process called protein synthesis. Two big steps, transcription and translation, like I talked about. So transcription is going from DNA to mRNA, while translation is going from that mRNA to an amino acid sequence. So first one we're going to talk about is transcription. And in transcription, what actually happens is that the DNA double helix is going to open up. Transcription is the first one that we're going to talk about. And what happens in transcription is that the DNA double helix opens up, and a gene, it only exposes one gene for a specific protein. And it opens up, and an mRNA molecule is formed based on that information. So basically it's one gene at a time. DNA is not transcribed as a whole entire molecule. It's transcribed in little pieces, single genes. So how this happens is that the RNA polymerase, so we had DNA polymerase that builds DNA molecules in DNA replication, but RNA polymerase is going to build an RNA molecule, all right? And very similarly, by forming nucleotides that match up with an open DNA space, and they match the complements AU and GC. And then once it gets to a stop signal, the mRNA molecule is going to be released, and we can see here that this is a complementary mRNA molecule. It's going to be released and let go. The DNA molecule isn't changed, winds right back up, and the mRNA leaves the nucleus through a nuclear pore. Then we move on to translation. In translation, the mRNA, which remember, DNA is read in triplets, so that means mRNA is going to be read in triplets. All right, but the mRNA triplets are referred to as being codons. Right? And those codons are going to play a role in how we build that sequence of amino acids, because the tRNA, the transfer RNA, contains the anticodon, which is the complement to the codon. And that complement is going to help make sure that the proper amino acid reaches the proper area of genetic information. So mRNA moves to the ribosome, where it's going to be read for translation in order to turn into an amino acid sequence to build a protein. And the tRNA carries a specific amino acid. That specific amino acid is going to be based on the anticodon sequence that will match up with the codon sequence. So the mRNA and tRNA are going to bind together, and then the neighboring amino acids that are there will form a peptide bond. Right? We should know that peptide bonds are what are between amino acids, and that will create the protein chain. So this may seem kind of a little strange to you, but if we look at this picture, here we have our mRNA molecule, right, being read by the ribosome. That's that whole entire thing, right? And what's actually happening is that this is our one tRNA molecule with an anticodon, another tRNA molecule with an anticodon, and each one carries a specific amino acid. And then side-by-side amino acids will bond together. And then we're reading in this direction, sorry. The one that's finished will leave, right? We'll shuffle down, and the next amino acid will come into place. And now we have two amino acids side by side, and they'll bond together as well. Right? So that transfer RNA kind of makes sure that it's carrying the proper amino acid for the proper genetic information, the proper triplet, the proper codon. And what amino acid is carried is directly based on this particular table. Right? This particular table shows us what the codon sequence is on the mRNA. So if the mRNA is going to read A, it's going to end up giving us lysine. All right, so there's kind of a method to this. By reading the proper base sequence, the base sequence directly codes for specific amino acids. And back to this picture again, so it kind of shows us that in here, what's taking place is transcription, just like it shows, and then out here is where translation occurs. So transcription in the nucleus, translation out in the cytoplasm at the ribosomes. So the first thing that happens, it says DNA information is copied or transcribed into mRNA following complementary base pairing. Right? Then the second thing, mRNA leaves the nucleus and attaches to a ribosome. Translation begins as tRNA anticodons recognize complementary mRNA codons, thus bringing the correct amino acids into position on the growing polypeptide chain. As the ribosome moves along the mRNA, more amino acids are added. At the end of the mRNA, the ribosome releases the new protein. tRNA molecules can pick up another molecule of the same amino acid and be reused. So the tRNA basically just comes, drops off the amino acid, and then leaves and goes back to pick up more. Guys, this pretty much wraps it up. We will be having a benchmark on this information so that hopefully we can get a better idea of exactly what's going on. If you have any questions, please feel free to see me.